So we'll start with the lecture. This is the introductory lecture for Python for Biosciences, Bioinformatics, Chemistry, Biology, Pharmacy, and Life Sciences, the beginner's course. This is brought to you by STEMskills Lab. This course will be instructed by me. I am Kanav Kalra, and partly it would also be taught by Dr. Gorde from Texas Medical School. So in the introductory lecture, discussing about today's goals, what we would cover in this introductory lecture would be the basic course information, like what we are going to be studying in this course and what all dimensions of subjects it encompasses. And we'll also give you a basic introduction to the computers and programs and how we can use Python, like specifically Python in this course, in order to solve the problems related to biology, chemistry, and other life sciences and natural sciences. So moving on. The course goals are basically the introduction to computer programming using Python. And the primary target audience for this course basically comes from two spectrums. And these two are basically two opposite ends of the spectrum. The first group of people would be researchers in biochemistry, biotechnology, medical sciences, life sciences, pharmaceutical sciences, chemistry, who are interested in learning a programming language, whether to automate their tasks, whether to do some sort of analysis, or just to help them, accelerate their research. The other end of the people, the other group, would be researchers in other areas who are interested in using computers, who are already like a little bit well versed with computers to solve life sciences, chemistry, pharmaceutical sciences, and the problems related to biology and all the natural sciences. So these would be the two basic primary target groups for this course, and we try to bridge the gap between the natural sciences and computer science. Moving on, to give you an idea about the teaching format of this course, we'll alternate between the regular lectures and tutorials. The regular lectures would basically be, like just instructive lectures about the basic programming techniques, concepts, some concepts related to, biology, chemistry, and how those are applicable in programming. This would be the format for the regular lectures. And tutorials would be conducted in which we would discuss problems. We'll try to solve some exercises related to life sciences, and these would be more hands-on, and I think these would be the star of the show, since this is where you'll be practicing and you would be learning programming skills and trying to, apply the things you learn on the problems related to your fields. The whole course would look something like this. It would consist of 14 to 15 hours of lectures and 5 to 7 or 5 to 6 tutorials. Each lecture would be 1 hour long, in which basically the first 45 minutes would be teaching, and the last 15 minutes would be devoted to exercises or to discussing problems or discussing doubts if you guys have any. But this is like all in all what the whole course teaching format would look like. We have two options for this course. One could be the weekday batch, in which there would be regular lectures and tutorials from Monday to Friday, 7 to 8 p.m. Indian Standard Time. Or there's also an option for the weekend batch, in which the course would be taught on the weekends, on Saturday and Sunday. But the lecture length would be 2 hours, 6 to 8 p.m. So depending on, what suits you, you could, in the email at stemlabin, you could tell us like which course format would be applicable for you. Moving on. This slide should just give you like the usage of computer science to solve scientific problems. Like in this course, we are basically trying to use this. We are trying to use computer science, or the techniques or the tools or the programming technologies of the computer science to solve problems related to, any natural sciences, whether it's biosciences, chemistry, pharmacy, biology. Uh, the basic contrast between these is that in computer science, like we see, or you guys must have seen in the movies, everything is either 0 or 1. Like everything is either completely true or false, like, and that is almost always the case. But in case of biology, chemistry, or other sciences related to nature or other natural sciences, almost nothing is ever completely true or false. There's always some probability related to everything. Like you guys might have read in the research articles or even in the literature when you were doing your studies that people always predict things with some probability attached to them, and it's never completely a finite statement. So the contrast that comes is that the biologists, chemists, and scientists, they seek to understand the complicated and the messy natural world around them. And computer scientists, they basically strive to build their own clean and organized virtual world in which they could control everything, and almost everything is built from scratch depending on the environment that they want. Moving on. The whole point of using computers or computer programming or the programming skills to work with natural sciences basically comes because everything in this day and age is about data, and data is key. Like data is originating from experiments, from observations in like biology, people doing their observations and experiments, and chemistry from chemical reactions and other chemical processes that people do. Like everything generates data, and data gets collected from everywhere, and this is basically where the computers and the computational power could help us in analyzing things and coming to conclusions. In the biology and the chemistry world, and even for other life sciences, the data is basically available in two formats. It's either available in the form of sequences or in the form of structures. And sequences are stored in sequence databases, and you know like similarly structures are stored in structural databases. So once we have the data and once we know what problem we want to solve, we could use the computational power of the computers to solve those problems easily, even if the data becomes huge and impossible to handle in a human manner. So once we know what we want to do on that data, once we have the logic that we want to operate on that, we could build algorithms for that, and then we could write the programs that could do the same thing, and then we could basically harness the computational powers of the computers to do that task for us. But the basic idea is that if your problem is defined, if you know what logic to work upon, you could do any sort of thing, and you could command your computer to do any sort of thing which could automate your tasks or which could help you make the analysis or conclusion in an easier and a faster manner. Do we have any doubts till now, or shall we move on? Okay, so moving on. We just talked about like data in the world of natural sciences and life sciences. It's either stored as sequences or structures. So in the term of sequences, the sequences are stored in sequence databases. There's Entrées, and Entrées covers over 20 databases, including the complete protein sequence data, which comes from SwissProt, UniProt, PDBs, EBI, and it also contains nucleotide sequence data, which comes from GenBank, that includes information from EMBL, DDBJ, and other sequence databases. So the sequences are basically stored, like the genetic information is stored in the form of sequences, and it's stored for the DNA and RNA, like the nucleotide sequence data. Like here you could see in the example, if you guys could see the circle, if you guys could see the cursor, you can see here like this is the sequence data available for putative promoter sequence, but for now let's just say that it's present here for some random thing. So this is the sequence data. It's for the DNA of it's for the DNA of this particular thing, and we know the DNA sequence data is made up of four nucleotides, A, C, G, and T, the four alphabets. And here you could see that like it starts with CCA, CA, CA, CA So if you have to do any sort of computation on the sequence data like this, if the sequence is like small enough, it's possible to do it, either by looking at the data or either by, doing calculations by hand. But if the data becomes huge, which it usually is in most of the cases, like the human genome we know is made up of like billions of base pairs. So doing the computations on that, like by hand or, by just doing the calculations on your own becomes humanly impossible. And that's where the computation could help us. And so like for a very preliminary analysis that could be done on the sequence data, it could just be counting the letters in DNA strings. Like let's say we have some DNA string that contains letters A, C, G, and T that we just talked about. It's made up of these four bases that make up the DNA. Like somebody could ask you the question like how many times does a certain base occur in the DNA string? Like let's see for the example given here, if the DNA is ATGGCATTA, we can ask how many times does the base A occur in this string. Like because this is small enough, we could just, go through each of the nucleotides and count, okay, A comes once, twice, thrice. So we see that A comes three times. And similarly, if somebody asks how many times does T come, it could be one, two, three again. And similarly, G comes twice and C comes once. So like these are like one of the very basic analysis that people do before jumping into the more complicated ones. And these could be done using the computers when the data becomes large enough. And another thing could be the frequencies of the bases A, C, G, and T. So to compute the frequencies of the bases A, C, G, and T, that is the number of times each base occurs in the DNA string. It's basically the count, the number of occurrences of that nucleotide divided by the total number of occurrences of all nucleotides. So let's say, for example, if the DNA string is ACGGAAA, we know the total length is 1, 2, 3, 4, 5, 6, 7. And we could see that A appears 1, 2, 3, 4 times, C appears 1 time, G appears 2 times, and T appears 0 times. So we could say that the frequency of A is 4 by 7. Similarly, C appears once, so its frequency is 1 by 7. G appears twice, so its frequency is 2 by 7. And T does not appear, so its frequency is 0. So these could be some of the very basic problems that could be asked about the DNA sequence or any nucleotide sequence data. And from a coding perspective, like we could do this very easily. We could create a function for counting how many times each base occurs in the string. And then we could create another function for computing the frequencies. Basically, this would be dividing the number of occurrences by the total number of occurrences. In both the cases, we want dictionaries such that we can index with the character and get the count or frequency out. Like in this case, a dictionary is something, it's a data structure in Python, which is something like basically like the dictionaries that you use in normal life. It's just that in this case, the dictionary is context dependent. Like because in this case, like the thing we want related to the bases is their count or their frequency. So here there's something called key value pair in which the base would be the key, like the character of the base would be the key, and the count or the frequency would be the value related to it. Here we could create a function like this is a function get base counts that's created. It gets a DNA sequence data string, and then it basically does the counting of like the number of occurrences and the frequency for the DNA string. It uses programming constructs like conditional statements and loops in Python to complete the following task. But like once you know these concepts, you could build these functions very easily. And then it does not matter if the DNA sequence is small or like super large, the computer would do the work for you once you define the logic on your own. Moving on, like here you could see we have a DNA string that is long enough, and let's say because we wanted to see the frequencies of the in the previous slide we wanted to see the frequencies of the bases. Here we are just plotting that in a beautiful manner to give us an idea like which base is the more dominant one in the whole DNA sequence, and this could give you an idea related to the problem that you're trying to solve. And similarly, these things like these plots could be created very easily with Python. Like we just basically create a list here that shows us, and we store the bases in it. We create another list that shows the percentage of how many times does it occur. Then we just use this. There's a thing called libraries in Python. We use the library Plotly to create a bar graph very easily on this. We give it different colors, and here we show the percentage on top of it. Like this becomes very easy to use also, like because if you see this in live action, if you hover over these bar charts, their percentages explode and like their fonts become large. So that makes the analysis very easy when the graph gets complicated enough. And so, like, just to give you an idea, these sorts of problems could be solved very easily with just simple lines of code in Python. Like, you could use the concepts like lists, plots, like in the previous slide, we saw the conditional statements and loops. All these constructs, if combined properly, they could help you like come to an analysis, a conclusion, or help you with that easily. Moving on, like similarly, we like those pre-processing or like or those calculations were done on the data that was small enough, but usually the data is very large, like we just see here, and it's stored in the form of FASTA files, which contains not just the string, but it also contains, these characters that tells us about which DNA sequence does this string belong to, and like what are the characteristics of it. So Python could also help us pre-process the file, like what's mentioned here. Like in this, the pre-processing would help us remove these extra characters so that the computation that we do just happens on the DNA sequence that is needed, and it does not include any additional characters that are not part of the sequence. And so all these things are very easily doable with Python. And once you create like the functions or tasks for that, you could do it in any sort of sequences, and you won't have to write the code again and again once it's done. Moving on. But there's another famous problem in bioinformatics and biology, which basically is about like after the preliminary analysis of getting the counts and the frequencies and like which base is dominant, there's another question about what is the GC content of the sequence data. And this thing, GC content, is relevant and it's important because like we know in DNA, it is made up of four bases, A, T, G, and C, and the bases A and T make a base pair, and the bases G and C make a base pair. And the thing is that the base pair made by GC contains three hydrogen bonds, whereas the base pair made by AT, it contains two hydrogen bonds. So the more GC base pairs are present in a DNA string, the more hydrogen bonds are there, and hence the stability of the whole DNA sequence is more. So we could easily solve the problem of GC content with just simple lines of code like given here. We have the DNA sequence here. We keep the count of AT and GC 0. Like here we see the conditional statements, like if bases A or T is encountered, it increases the count of AT. If it encounters G or C, it increases the count of GC. And basically then it counts the percentage of dividing just the count of GC divided by the total count. And here we could see like for the DNA string here, the GC content of the input string is 41%. And so like you could do these sort of analysis very easily. And then again, it could help you like in the further downstream processes of whatever you're trying to do. Moving on, there's another famous problem of reverse complement. Like once you have a DNA sequence or once you have an RNA sequence, people are usually interested in finding the reverse complement of it. And then again, we could create a very simple function like reverse complement in which it takes a DNA string, and then again it creates a dictionary. And in this dictionary, the context is basically for A we want T. These key value pairs would be for A we want T, for T we want A, instead of in place of G we want C, and in place of C we want G. We do this sort of operation for like we go in the loops, we do this operation for each base in the DNA string. And then once we have a DNA string, we give it to the function, and the output that we get is the reverse complement of it. So you could see like these problems, which are like which were major problems back in the day, which are still given as exercises in the the biology courses taught in undergrad and courses, in which people have to solve it by hand. Like these are very easily doable if you just code it in just a few lines of code. Moving on, I'm pretty sure we all are very much aware with the central dogma of life, and we all must have studied this in high school, in which we know how the genetic information is passed from one generation to the other, and how the characteristics are imparted from parents to the children. Like basically, the DNA data, the genetic data is passed from the parents to the children in the form of DNA, and then there is DNA replication, which makes DNA from DNA, and DNA gets transcribed into the RNAs. This process is called transcription, and then the RNAs gets translated into proteins, and this is the translation. And these proteins are basically responsible for the characteristics that the individual organism shows. And so this is basically the whole central dogma of life, which has been the study or the point concept of research for like decades now, and like people have like it's relevant in bioinformatics, biology, even somewhere in chemistry it's relevant. And you could see that this like people could ask sample problems, like they give you a DNA sequence, they could ask the problem like transcribe the following DNA sequence and then translate the resulting mRNA sequence, and ultimately what protein sequence would you get. Like this problem, it's essentially in itself a whole, it's the whole concept crunched into one single problem. And if you have to solve it, like on paper it's slightly difficult, but like we could solve it easily with Python if you know the logic and like what you want to do behind it. So here, like we say, we have a human gene for like casein kinase 2 here, and we have the DNA sequence. First, we have to know where in the DNA sequence are the genes located. And basically, just to give you an idea, once you have a DNA sequence, there's a thing called start codon and there's a thing called stop codon. And we know that whatever starts with the start codon and comes before the stop codon, the thing that's stored inside these two, basically makes up, this is where the genes are located. And there's a table called the codon table, in which basically, you know, three bases, they match up to a codon, and it could either be just the same, like there's a start codon and there's stop codon, and whatever comes in between, it's used. It's basically the genes, and then those genes you could create the peptides or the amino acids depending on the sequence data that it has. And we could code this easily in Python if you want. Like we could create, like if you know the logic what to do, you could create a function called translate that could, or like then you could also create a function for transcribe, like for both transcription and translation. And like I'm just showing you here a screenshot of like what I did to code these things. I'm just showing you one part of the logic. And the idea basically is that you could solve any sort of these problems. Once you know the logic behind it, you code it, and then you can perform it on multiple sequences or like any length of sequences if you want to do it. Like here, if we have the DNA string like this one here, then basically this function, I have this one function translate that would translate the whole DNA sequence into the proteins. So here we see that there is a protein sequence, like these are the possible protein sequences. And here in the previous slide, we saw that like this methionine is the start codon, and these ones are the stop codons. So here we see that in this one, this protein sequence starts with M and stops at the stop codon. So these are the possible protein sequences that are, you know, possibly translated from this DNA sequence data. So all these problems could be solved with Python if you know the logic, and once you learn to code it, it becomes very easy. Like what we saw was just for the nucleotide sequence data. We could do similar sort of operations on the protein sequence data. It's just that, in the nucleotide sequence data, the letters were just A, T, G, and C. In the protein sequence, they become much more. So like the questions could be similar. Like first we asked about how many occurrences of each of the bases was there in the DNA sequence data, and similarly they could ask how many distinct amino acids are present in the protein sequence, and similarly they could ask what is the percentage of each amino acid, which was just like the same as frequency. And like once you know like which amino acids are aromatic, you could also tell like what is the percentage of aromaticity of the sequence. And like once you know what are the charges on each of the amino acids, you could also tell that what is the percentage of positive or negative charge on the whole protein sequence. And these sort of operations could be done very easily with the concepts of loops, conditional statements, lists, dictionaries. Once you know these, you could code these problems easily. Moving on, there's a very famous problem of sequence alignment in biology and bioinformatics. And sequence
Sorry, the voice is not clear. Sorry.
can you hear me now?
Sir, we can hear you, but the skin is excess zoomed, so we cannot see the skin properly.
Okay, let me just see. Can you guys see the screen now?
No, sir.
Okay, okay. Let me just.
Yes, sir.
Can you see the screen?
Yes.
Yeah.
You guys can see the slide of sequence alignment?
No, sir. Only STEM.
Sir, I can see the only logo, but I cannot see the slide.
Oh, and was it happening like this whole time?
No, sir.
Okay, okay, okay. Then let me just, change the screen sharing setting and can you guys see the screen with the like Google Meet window?
Yes,
Yeah.
sir.
Okay, okay, okay. Perfect. Then I'll just, just display my entire screen. Uh, Let me know. So this is visible now?
Yeah.
Is it still visible? You could see the slide with sequence alignment?
Yeah.
Okay, perfect, perfect. Like, thank you for stopping me. I did not know it was happening like this. So, I'm assuming that all of us can see the slide. So now we'll just move on what we were looking at. There's like a very famous problem of sequence alignment in the fields of biology and bioinformatics, like particularly in bioinformatics. And sequence alignment is a method that is used to compare the similarity among the sequences. The nucleotide sequences are composed of four letters A, C, G, and T, what we just saw. And like the sequence alignment is done to see like if two sequences are similar, and which further helps them to see like if their functions would be similar, or like if they come from the same, genetic lineage. Like for example, let's just consider the following sequences. There's this first sequence S1 ACAGTAG, and
Sir,
there's
again, we cannot see the slide.
sorry,
Sir, the skin is not showing.
the screen is not showing. Okay, let me see what's happening actually.
Sorry, sir. Can you make it full screen, please? Thank you.
Uh, is it full screen visible now?
Now it's
Yes, sir.
okay.
No, it's
Yes, it's visible.
no,
Okay, okay, okay. So we were just talking about sequence alignment and like how sequence alignment is like how is it relevant, and like how we could do it in Python. So like let's say consider the following these two sequences, and like what we could do to find the similarity among these sequences is that we could try to find the similarity among these visually by just placing them one over the other, but we might miss the best possible alignment in this way. and also this would become like cumbersome and humanly impossible when the sequences are fairly long. So we there's already a standard procedure to find the best possible alignment among two given sequences, like in which there's an algorithm that's already available in the literature, in which they try to align the characters of sequences, both the sequences one by one, and there are three possibilities. Like if the characters in both the sequences are the same, it's a match. If the characters are different, it's a match. If it's a mismatch, and if there's a character in the first sequence but there's not a character in the second sequence, it's called, or like there's a character in the second sequence but it's like missing in the first sequence, it creates a gap. So we could like using this algorithm, like for these two sequences, there's a concept of global alignment and local alignment. And in global alignment, they try to align the whole sequences one over the other. And in local alignment, they just try to align the best possible part that aligns of these two sequences. Like here in the example, we could see that this S1 sequence ACAG it is aligned over the second sequence TCAG in which the first one here is a mismatch because the characters are different. Like next four characters are matches, and then in place of A, in the place here in S1 it's A, in S2 it's nothing, so it's a gap, and then again it's a match at the last. So this is like the global alignment for these two sequences, and the local alignment gives us the CAGT. You like once the algorithm is known and once you could create the logic, you could like this is what I did to code this in Python. I wrote the logic and basically gave it the two sequences, and it gave me that output as the answer. Like this all is also possible with Python. There's another thing very beautiful and like very powerful with Python is that it contains a lot of libraries, packages, modules, and these are basically the things that are already built by people who have used Python and who are from, the fields related to something that you might be doing. The thing is that the community is so huge. So there's a possibility that if you're trying to do something with Python, there's a possibility that either that thing or at least some part of it would already be, coded, implemented, and available somewhere on the internet. So like if you're trying to solve problems related to biology and biochemistry or even some part of chemistry, there's a package called BioPython available in which these things are already built. Like there are already functions built for that. Like here to do the sequence alignment with just basic Python, I had to write this whole code, and this is not even like the full code. This is just a part of the screen. This is just a screenshot of some part of it. But in BioPython, like here, we could see that there are already functions built. Like here you have bio, we want to do pairwise alignment, whether you want to do local alignment or global alignment, you could just do it in one line. So this is one of the beauties of Python because a lot of content has been built for it and it's supported by almost people from all the scientific backgrounds. So here, like what we saw as the sequence, like for the global alignment and the local alignment, this program gave me the same output. So this just gives you an idea, like once the logic is known and the logic is consistent, and like if the packages are also known, it could make your life very easy as well. Moving on, I think we've talked enough about sequences and like what sort of analysis is possible on sequences and how easy it could make the conclusive part of the research. The next thing we talked about was like the data also formed in the form is also stored in the form of structures, and data is stored in structural databases. And this is more relevant to biologists, chemists, life people from life sciences, medical sciences, or pharmacy as well, because it contains the structural data about biomolecules, which encompasses DNA, RNA, protein, glycans. It contains data about ligands, drugs, ions, water, and all the other chemical molecules possible. So the structural databases for small molecules in this case is the Cambridge Structural Database, CSD. It contains data about drugs, agrochemicals, pigments, explosives, protein ligands, and it basically contains data for the small molecules. But then again, this is much more, relevant for the people who are either from the medical or pharmacy background. like this structural data would mostly contain the molecules that you would be interested in. Now, the beauty about the Cambridge structural database is that like the data in it, like here you could see that it's growing at an exponential rate, like almost every day with the higher computing power, more and more experiments are being done and more structures are being resolved and stored into this database. Like here you could see that more than 50,000 new structures are added each year, and you could see that the growth is exponential. So once all this data is available, people have to do some sort of analysis, some sort of conclusive analysis on this to, to make use of that data. Otherwise, it's just, something that's stored somewhere in a hard drive, which won't be any use to anybody. And another database where the structural data is stored is like PDB, which is for biomolecules. The PDB stands for the protein data banks. It's primarily for biomolecules. Here you could have the structural information about the DNA, about the RNA, or the DNA-RNA hybrid, or even the DNA protein complexes. You could have all the data, structural data about these these biomolecules, or even if you are, aware of the coronavirus, COVID, which I'm pretty sure all of us are, and how is it has affected all of us. It has also contained, like, it's so up to date that it also already contains like the protein structures for the COVID-19 virus spike receptor binding domain. Like it already has structures resolved related to the COVID-19 as well, to give an idea like how fast things are moving. And also for the PDB database, like here you could see that every year more and more structures are being added to it. And like even in just 2020, probably because of COVID, like there was a huge jump in the number of structures stored in the database. Like, and even in 2021, even though it's just been January, more than 600 to 700 structures have already been resolved and added. So this could give you an idea like at which pace the data is being generated. So people need to do some sort of analysis on that to make use of it, or to make sense of what they are already, generating. Like moving on, because we know that the structural databases are stored in these databases now. The structural data is stored in the databases now. It's basically stored in the PDB format, which is the protein data bank format, and it stores, like, this is the, star of the show, the coordinates. The coordinates basically, which tells like at which point which atom is located. Here, like this row could tell you whether it's a atom or heteroatom. Like here they tell you like which element is it from. Then it also tells like which amino acid it belongs to. Then there's a chain number, sequence number. All these things are available for each of the, atoms present in the biomolecule. But this is where it shines. Like these are the coordinates that, you or I would be much more interested in using, and this would be like the core of the analysis. It contains the XYZ coordinates, which basically tells you like where in space that atom is present relative to the other atoms in that biomolecule. And this is what we would be relevant for the biologists, biochemists, chemists, pharmacists, and people from these backgrounds, because they want to, do some sort of analysis on the structure of the molecules related to the problem context that they are trying to solve. Like here, just to give you an idea like how the coordinates are stored and like how the representation is done. Here on the left you could see it's a benzene ring, C6H6. We all learned about this in high school. It contains alternate single and double bonds, or here just you could just show it by resonance here. Like it contains, or with every carbon there's a hydrogen attached. That's how it's C6H6. It's a planar molecule. And here I've mentioned like which, this is like the C1 and this is the H1 related to it. And the coordinates corresponding to it are stored here. Like you could see like it contains C, CH, CH, CH for each of the carbon and hydrogens. And these are the coordinates for the C1 and H1. These are the X, Y, and Z coordinates. And once you have these coordinates stored, and once you have these coordinates available for the analysis, you could do any sort of operation on it that is relevant to your problem. So this is just basically how these coordinates would be represented in plain files. Moving on, like once you have the coordinates of a benzene ring, you could do any sort of mathematical operations on it. Like Python can be used for various translations and rotations to get the desired structures. Like once you have one benzene ring coordinates, you could create any of these structures. Like you could create a sandwich, a T-shaped or a parallel displaced benzene ring, in which what you just have to do is you have the coordinates of one benzene ring, you copy those coordinates for another benzene ring, and then just place it at what distance you want on top of it. And then you could just like this would just be like a simple translation, and you could create a sandwiched benzene structure. Like if you want to do T-shaped and you have the coordinates of just one benzene ring, you could do the translation, and then you could do a 90-degree rotation, and you could easily create a T-shaped a T-shaped structure for benzene. And similarly, these would contain two translations, one in the x-axis and one in the y-axis, and this you could create the structure for the parallel displaced benzene ring. Like all these operations are also available like in a multitude of software that are available, but the possibility is that like some of the softwares are very restrictive, or either they are paid, and then again there is a possibility that because you guys would be interested in doing the research on those, so you might be wanting to do like some sort of manipulation on this benzene ring that might not be available in the software. So like once you are well versed with like how to do these things in Python, you would be beyond you like the possibilities of what you could do goes beyond those softwares, and you could do any sort of operations on these. Like moving on, like here we show you a water molecule. It's H2O, which contains of two hydrogens and one oxygen atom, and like the OH bond length is around 0.9 angstrom, and there's like the HOH angle is around 105 degrees. So like then again, like there are softwares available in which you could create a water structure easily, but then again, like if you want to create something instead of water or something more than that, then there's a possibility that it might not have it, but it's very easy to do once you know the logic behind it in Python. Like to create a water structure like this in Python, I could easily just place this oxygen at some coordinate, let's say 0, 0, 0. Then I could make a line segment, OH line segment, in which I know the length is this, so the coordinates of this hydrogen would be known to me. And because I assume like we know mathematics, then we could easily use the sine theta cos theta concepts to find the coordinates for this hydrogen. And like once you know like how to use these small mathematical operations, you could do any sort of structural analysis that could be relevant to your field of work. And like this should just give you an idea of like how awesome and how powerful Python is, even in the case of like structures as well. Moving on, like this is like something that is much more relevant and much more related to people working with life sciences, medicals, and pharmaceuticals. Like here we show you the usage of artificial intelligence in drug discovery. So here like initially we talked about it's everything about data. Here we have the data of the molecular log p, molecular weight, the number of rotatable bonds, the aromatic proportion, and this is like the log solubility value of those molecules. Like here we just they try to build a model in which they could predict the log solubility value from these given values here. Like these values are called the descriptors, and this is called the target value, in which you could use once you have either the structure of the molecule, or like once you have the structure of the molecule, you could calculate these descriptors from it, and then once you have a model built, then you could easily predict the log solubility value on top of that. Like here we see that we created a model, something using something called random forest, in which it basically gave us the equation of how to, calculate the log solubility value using these descriptors, and these descriptors were computed from the structure using a package that's already available in Python. And in the next slide, we could just show you like how this is done. We created the model, and then we wanted to see like how accurate our model is. So this is something like a correlation graph in which we plot the experimental versus the predicted log solubility value, and we see that it's pretty accurate even though the data we had was not huge. So if we have more data, we could do this more easily. And this is like one of the finding the log solubility value of a structure is like one of the major problems that comes in the field of drug discovery. So all these things are possible once you're like well versed with Python and you know how to play with data, which we hope that if you take this course, you would get some idea how to do it, and then you could be on your track to, do your research in a much more fun and easy manner. And now we tell you what the course syllabus is. Basically, like in the initial introduction lectures, we'll give you an introduction to biosciences, biology, chemistry, and the introduction to computation and what programming is. Then we introduce you to Python. We tell you what it is and how to install it. Then we learn more about Python and try to get our hands dirty with it, the basic input output, mathematical operations, conditional statements like we saw in the slides. Then we tell you about the data types and iterable objects like the list, strings, tuples, dictionaries, like some examples we saw in the slides. Then we tell you about loops, like how to do the same things again and again with just simple codes, and then how to deal with the data that's present in files, whether it's text files, PDB files, or sequence FASTA files. Then we tell you about how to write functions and how to use modules and packages to personalize your code. Then we introduce you to sequences and like help you do some preliminary sequence analysis, like introduction to sequence, exercises on it, percentage, GC content, reverse complement, like all that we saw. Then we also introduce you to Jupyter Notebook and Jupyter Lab and even Deep Note, which is a more research-friendly environment in which you could, see your analysis right there whenever you perform it. Then we introduce you to the popular packages with the scientific community, like NumPy, which is used for doing mathematical operations very fast on huge data. We introduce you to Matplotlib that creates beautiful graphs with the help of Python programming language. We also introduce you to Pandas, which basically helps save the data in a database in a format that is like much more uniform and easily readable as well. Then we introduce you to BioPython, like we just saw. BioPython contains a lot of functions. It's a huge library that might already contain whatever you are trying to do. Then we tell you how to do sequence analysis with BioPython, like similar exercises what we saw, but now with just much more ease. Then we introduce you to sequence alignment and how to do it in Python, like what it is. And then we introduce structural analysis, like introduction to the PDBs, how to read and write PDB files, how to distinguish like these atoms, head atoms, how to find the distance between the different atoms in a molecule and a biomolecule, what's the angle, what's the torsion angle, how to get the relevant molecules out of the big huge PDB files. Then we introduce the concept of phylogeny and the phylogenetic trees and how it could be built and used in Python. Then we give you a flavor of the cluster analysis of the machine learning in Python and how it might be useful in biology and chemistry, like using the unsupervised machine learning, supervised machine learning. Then we also give you some more tips about like some new libraries or like how to create meaningful graphs and like how to do the graphical interpretation in a better manner. So this would be like the gist of the course syllabus. And the tentative start date for this course would be 8th February and the timing would be 7 to 8 p.m. Indian Standard Time, using the weekday batch thing. Uh, and like if you have any queries, you can ask them now, like any questions, or like if you have something that is much more specific and you want to ask us later, you could always drop us an email at stemlabin@gmail.com.